And now I request Dr. Shyam Prasad to give the next.
I thank the organizers for giving me this opportunity.
Excuse me, sir.
Yeah. So my presentation is about pigment, microbacterial pigment. My presentation is divided into three parts. One is isolation of bacteria and identification, production of pigment and its applications. So I'll be taking 5 minutes. So today textile industry is making a business of around 9 billion US dollars with an average dye consumption of 8 lakh tons. You know, these dyes are toxic, they are carcinogenic, and they are released into water bodies. So they pollute groundwater as well as they contaminate water bodies, and they accumulate in aquatic life. From aquatic life, they indirectly enter into human beings and pose health issues. So in this way, textile waste is discharged into the environment. So here, the industrial people are looking into natural colors because natural colors are eco-friendly, they are stable, and among natural colors, microbial pigments are very stable as well as they grow on cheap media and their generation time is very less, and also ease of genetic manipulation. So here we can see some of the examples of pigments already available in the market. So these are the pigments and these are the applications. So in our study, I have selected prodigiosin. Prodigiosin is a red pigment which has many biological activities like anti-cancer, anti-malarial, the list goes on. So despite many biological activities, so the application is very limited because of high production cost. So researchers are working to reduce the production cost. So this slide shows the suppliers of prodigiosin. So on average, the price of prodigiosin is 25,000 for 1 milligram. Okay. So here we have isolated an organism capable of producing prodigiosin. So this is the slide which shows isolation. And this is the prodigiosin pigment. So this is prodigiosin when plated. So the bacteria which we have isolated, identified as Serratia surfactans. So we have deposited in gene bank with the accession number. So then we have taken some synthetic media from literature and we have gone for production of prodigiosin. So we could get peptone glycerol broth is giving very good results of 5098.5 units per cell. So this is the yield of prodigiosin in synthetic media. Then we have used agro waste to reduce the cost. So when we have used agro waste, this waste we got from oil mills after oil extraction. This is no use for anyone. So here, by using solid state fermentation, we have gone for production of prodigiosin. So among different agro waste we used, so this black sesame is giving very good results. Like, so peptone glycerol broth is giving 5098 units, whereas black sesame is giving 69,000. So this is the difference of using agro waste. So here we can cut down the cost. So here we have gone for mass production. We didn't go for scale up. We gone for mass production. So here this is the pigment after extraction. So after purification and all, we got the pigment. So then we went for a structural elucidation by using different spectra. So this is structure we got. Prodigiosin has got different derivatives also. So we got, this is the structure. Then we have also used for synthesis of nanoparticles. So by increasing the concentration, we got nanoparticles also here. So this is the UV spectra of that. So then we went for application. So here we went for dyeing of different fabrics here. So in the first row, we have selected different fabrics. First row is application of only prodigiosin, and second, third, fourth, fifth row is we have used different binders. So when we went for washing, so without binder, we lost the color. And where binders are applied, the color is retained here. So we also tested antimicrobial activity of this dyed fabric. So you can see, against clinical isolates. So we got almost 90% reduction in bacterial growth. Okay. So this is antifungal activity, 50% reduction in fungal growth. So these are some pictures. And also we have studied in silico study, anti-eczema activity by using in silico studies. So this is atopic dermatitis. Prodigiosin is showing good results against atopic dermatitis also in silico studies. So by using this prodigiosin, we can also use for designing of antimicrobial shirts, underwears, moss for controlling of skin infections. Not only for dyeing, not only can be used as a dyeing agent, but also can be used for designing of antimicrobial fabrics. Then we also designed antimicrobial soap by using only prodigiosin. And when we have tested this, it is also giving very good results against bacteria as well as fungus. So these are the results. Then we also gone for Band-Aid. We also prepared Band-Aid by using prodigiosin silver nanocomposite Band-Aid. So by using nanoparticle prodigiosin nanocomposite, we have prepared Band-Aid. This Band-Aid is also working good against clinical isolates, bacteria as well as fungi. So control Band-Aid has not given any inhibition, whereas our prepared Band-Aid is giving very good results. So this is a picture. So we also prepared prodigiosin nanocomposite ointment. So this is ointment. So this is also giving very good results against clinical isolates of bacteria as well as fungus. So we also prepared Band-Aid. So this can be used for infections of the wound. So scope of prodigiosin is we can prepare antimicrobial fabrics, we can prepare Band-Aids, bandage, ointment, prodigiosin soap. So we recently have seen prodigiosin is also inhibiting viral replication, that is herpes simplex virus. So we can also prepare eye drops to control this virus also. So we are collaborating this micro tech company for further studies. Thank you.